This video provides an explanation of the Lac Operon. Bacteria, in this case E. coli, are very efficient. They don't make proteins when they're not necessary. And we're going to talk about two proteins called permease and beta-galactosidase. These two proteins are necessary for E. coli to take in and break down, digest, if you will, a molecule called lactose. And the permease is one that's going to be a protein to allow the lactose to get into the cell. And then galactosidase is going to break this covalent bond that's in between the two monomers of this disaccharide called lactose. Those monomers are glucose and galactose. Once that breakdown has occurred, then glucose is available for fermentation like we studied previously. Now, in a normal state, let's say a lactose-free state, meaning that there's no lactose available for E. coli to digest, it looks like this. There's some permease, there's some beta-galactosidase, but not much. When, as we see over here, lactose becomes available, that lactose, I will describe, is going to move into the cell and not only be broken down, but it's going to turn on expression of more permease and more galactosidase so that we can see the cell after lactose is present has many more of these purple permeases and much more of the green beta-galactosidase. This will involve something that is a repressor and a small effector molecule. So let's look to the right-hand side first. Right? Normally, RNA polymerase will sit down on a promoter and transcription will occur. And here we have mRNA that's being made. When repressor molecules, effector molecules like a repressor, sit down around the promoter region, they don't allow the RNA polymerase to get to where it needs to be. And so no transcription is made, right? No, is done. There's no mRNA that's made. The other molecule that we want to point out here is something called a small effector molecule. Lactose is an example of that. Lactose will bind to the repressor and it will prevent the repressor from binding to this area on the DNA. And so therefore, when lactose is around, the RNA polymerase can express genes and make mRNA. So what is an operon? This in particular is called the Lac Operon, and it's a big string of different genes and promoters. And so I want to first point us out to particular genes we've already discussed. We talked about beta-galactosidase and permease. I'm not going to talk about transacetylase. We're just going to ignore that one for right now. This gene is called the Lac I gene, the Lac I gene. I don't know why, but that's what they're called. Genes, as we discussed before, need to have a promoter for them. So here is the promoter where RNA polymerase is going to sit down and move through these genes to make mRNA, to transcribe the gene. An operon is really a group of genes, right? And so it is unlike the way we do it, where we have one promoter for one gene. They have one promoter for multiple genes. There's another little portion in here called the operator region. It's a piece of DNA that the repressor is attracted to. Now let's move up a little bit further. I have another gene called the Lac I gene, which actually makes the repressor. And then I have a promoter for that gene. So I have two promoters. One promoter is going to allow RNA polymerase to sit down and make the repressor. The other promoter is going to allow RNA polymerase to sit down and transcribe these three genes. And the operator region is where the repressor sits. So let's watch this in action, sort of action. In this case, I don't have any lactose available. As I mentioned, RNA polymerase is going to sit down on this Lac I promoter, and it's going to make the repressor mRNA, which is then going to be translated, and that repressor protein is going to sit here on the operator. This is something that's called constitutive. It happens all the time. Now, what you'll see happen now is this another RNA polymerase, or maybe the same one, tries to sit down at this promoter because he wants to transcribe these 3 genes, but he really can't get past the repressor that's bound to the operator. And so no mRNAs are made for Lac Z, Y, and A. We get no protein for those. Now, the next situation is when lactose is present. So I mentioned to you that the Lac I gene is always turned on. It's constitutive. So this repressor is made all the time. But now I want you to imagine as it's on its way to get to the operator, it bumps into lactose. And lactose binds to the repressor, as you saw on the previous slide, and that changes its shape. And it doesn't really look that way here, but it does change its shape so that the repressor can no longer get to the operator. And now what you will see, so here's the repressor being tied up, kind of held back by the lactose. Now the RNA polymerase comes in and it can move through the operator. It can transcribe the mRNA for the Lac Z, the Lac Y, the Lac A genes, and we get those 3 proteins after translation. We will discuss further in class about how glucose plays a role in this. But the key to remember is when lactose is available, we turn on the genes which allow lactose to get into the cell and to be broken down into glucose and galactose. When lactose is not available, there's no sense in making those proteins because we don't need to let anything in and we don't need to break it down. So when lactose, let's go back, is not available, as we see here, the repressor is made, it binds to the operator, and it doesn't allow transcription of these 3 genes. I hope this helps to make the Lac Operon a little more understandable.